Okay, welcome to our first lecture on muscle tissue. Here we'll be exploring one of the few tissues in the body that's capable of movement. Muscle tissue's job is to maintain body temperature and to promote internal and external movement. It does that by converting chemical potential energy in the form of ATP into kinetic energy, the energy of movement. Muscle tissue is nearly half the body's mass, and it transforms ATP into mechanical energy and exerts force, and it comes in three types: skeletal, cardiac, and smooth muscle. Any prefix that you see starting with myo, mice, or sarco indicates a reference to muscle. Now, skeletal muscle is found in organs and attached to bones and skin, primarily either attached to skin and connective tissue beneath, or attached to muscle and bone, or attached to bone and bone. We control skeletal muscle. When it's under voluntary control, the elongated cells are termed muscle fibers, and they have vertical light and dark lines called striations running through them. It contracts very rapidly, it tires very easily, but it's very powerful, and it requires nervous stimulation in order to make movement. Cardiac muscle is only found in the heart. It is striated and can contract without nervous system stimulation. In fact, the heart has its own conduction system that's actually embedded in the walls of the organ that acts as a pacemaker that tells the heart when and how forcefully to beat. It's not under voluntary control, although it does respond to our changes in activity levels. You know probably that when you exercise or exert yourself, that your heart rate goes up, and when you relax, your heart rate goes down. But this is all the result primarily of a branch of the nervous system called the autonomic nervous system talking to the heart and either speeding up or slowing down its response to nervous stimulation. We'll talk more about this when we get to Chapter 18 in A&P 2. Smooth muscles found in the walls of hollow organs such as the stomach, the urinary bladder, and in the airways. It's not striated, and it can contract without any commands coming from the nervous system. Okay, it's capable of its own spontaneous contraction. It's under involuntary control, meaning that it's governed primarily, again, by the autonomic nervous system, that branch of the nervous system that operates without us having to think about it. And so this is a chart sort of running down the differences in skeletal, cardiac, and smooth muscle. This tells you the location. This tells you what the cells look like under the microscope. So study this. This is very important. This will come up both in lab and in lecture. You want to be able to recognize these tissues when you look at them under a scope, and also if you were to see them, say, in a figure or an illustration. This is simply a listing of the connective tissue components of each of these muscle types. We'll get into these in more detail as we move through the chapter. This tells you whether or not myofibrils are arranged in contractile units that are called sarcomeres, and this is sort of a detail of the microscopic anatomy of muscle in terms of some of the features that we're going to see when we shrink ourselves down to the level of a muscle cell and look at how that muscle cell is able to take information from the nervous system and convert it into kinetic energy. Okay. Chart continues talking about the extent of the sarcoplasmic reticulum in each of the muscle cell types. The sarcoplasmic reticulum, as we're going to discover, is essentially a chamber that holds calcium that is allowed to either release or sequester calcium depending on whether or not the muscle is in contraction. Gap junctions are essentially tunnels between adjacent cells that allow electrical communication. These are found primarily in cardiac and smooth muscle. Neuromuscular junctions are simply the area where the nervous system and the musculoskeletal system interact. Um, do the cells have individual neuromuscular junctions? Yes, if we're talking about smooth and skeletal muscle. Not so much if we're talking about cardiac muscle. And then how is contraction regulated? And in skeletal muscle, it's via what we call the somatic motor division of the nervous system, which we control consciously. And for cardiac and smooth muscle, it's primarily through the autonomic nervous system, which operates without our conscious awareness. Further information in the table includes the source of calcium for its release inside the muscle that will result eventually in contraction, the presence of pacemaker cells, the effect of nervous system stimulation on these tissues, and the speed of activation, whether or not the cells exhibit autorhythmicity, their response to stretch, and what type of metabolic pathway they use in order to generate their energy for contraction. So believe it or not, that one chart, that one table that we just went through, describes all the information we're going to cover in chapters 9A and B. So if you know this material, then you're in pretty good shape for the exam. So I would say star these 3 slides, know this material, and you'll be in excellent shape. So what are some special characteristics of muscle tissue that distinguish it from any other tissue type? One is excitability. This is the ability to receive and respond to a stimulus. Generally, the stimulus is in the form of a chemical secretion that comes from a nerve cell and sets into motion a series of biochemical reactions at the cell surface and inside the cell that ultimately result in contraction. Contractility is the ability to shorten forcibly when stimulated. Extensibility is the ability to be stretched, and elasticity is the ability to return to your original resting length after you've been stretched. Okay, so muscle tissue exhibits all of these. What are some important functions of muscle tissue? Well, the movement of bones and the movement of fluids, a couple of very important functions of muscle tissue, maintaining the body posture and position, stabilizing joints, and the generation of heat. On a cold day like today, I'm sure you've noticed that when you sense a decrease in temperature, you begin to shiver. Okay, we call that shivering thermogenesis, and that's one of the body's ways to try and maintain a constant body temperature. We can't let our body temperature get too high or too low, otherwise cells will die, tissues will die, and then ultimately you will die. Other functions include protecting organs, forming valves. Valve is basically a doorway into or out of something. It also controls the size of our pupils. That's the opening that lets light into the eyeball. And it's also responsible for the production of goosebumps when we're frightened or when we're cold. The idea behind the goosebumps is that it produces a layer of dead air between the skin and the environment, helps us to retain more heat. The response when we're frightened actually goes back to when we were covered from head to foot in lots and lots of hair, and when we would be cornered by a predator, the hair would stand up and make us look like a larger item and less likely to be consumed as a meal, and so it was a protective response. If you want to see this in action, go home tonight and scare a cat, okay? And you'll see that hair stand straight up. Okay, so we're going to start with skeletal muscle. Skeletal muscle is under our conscious control. It has striations and it has contractile units inside the skeletal muscle cells that are called sarcomeres. Each muscle is served by one artery, one nerve, and one or more veins. They enter or exit near a central portion of the muscle, and they branch through connective tissue sheaths to serve every single cell. Every skeletal muscle fiber is supplied by a nerve ending that is going to regulate its activity. If it's not, then that skeletal muscle fiber will eventually atrophy and vanish. There's a huge nutrient and oxygen need for skeletal muscle because it does a lot of work, and it also generates lots of waste. This is why we have extensive vascularization in muscle tissue. So this is just a look at skeletal muscle. The connective tissue sheaths that hold it together surround the entire muscle, also serve to attach it to bone, and we'll see that they surround fascicles and individual muscle fibers, those would be muscle cells, in order to hold the muscle together so that when it contracts it doesn't fly apart. Okay? So this supports the cells and it reinforces the entire muscle. The connective tissue sheaths are called in order from superficial to deep the epimysium. Okay? This is dense, irregular connective tissue that surrounds the whole muscle and will blend with fascia and is also continuous with tendons that attach it to bone. The perimysium, which you can see surrounding the individual fascicles, okay? You can see in the blow up here the perimysium, okay? And it's also fibrous connective tissue. And then the endomysium, which is areolar connective tissue that surrounds each individual muscle fiber, okay? So this is all designed to do one thing, to hold the muscle together. It also helps to transfer the force that the muscle produces when it contracts to the elements that it's attached to. For instance, in this case, to the bone, and also produce movement at the joints. Okay, skeletal muscle attaches in at least two places: the insertion, which is its attachment to movable bone, and the origin, which is its attachment to the immovable bone. And depending on the type of motion we're talking about, the origin and the insertion can vary. Okay? Attachments can be direct or indirect. Direct attachments, where the epimysium fuses to the periosteum of the bone or the perichondrium of the cartilage, are quite strong and durable. And indirect attachments are via connective tissue wrappings that extend beyond the muscle as a tendon or as an aponeurosis, which you could consider to be a broad, flat tendon. And so this is a chart showing you the structural and organizational levels of skeletal muscle. I won't go into each one of these. This is 9.1 in your book. But again, star this material, know it. We're going to go into some detail covering it today. Okay, now if we examine the microscopic anatomy of a skeletal muscle fiber, which would be a single long cylindrical cell, we find that it's between 10 and 100 microns in diameter and up to 30 centimeters long. So we need to get a sense of proportion for these dimensions. Notice that there are, or remember that there are about 2.5 centimeters in an inch. Okay, so let's write that down here. Get an idea of these dimensions. All right, so 2.5 centimeters in an inch. Okay, so if we do that math, you're talking, about 10 inches long for a single cell. And we're up between 10 and 100 microns in diameter. Now, what the heck is a micron? Well, a micron is 1 times 10 to the minus 6th meters. Okay. And there are essentially 1,000 micrometers in a millimeter. Okay, because a millimeter, so we'll set this is equal to a micrometer. Okay, at 1 times 10 to the minus third is a millimeter. Okay, and I'm going to ask, okay, well, what about centimeter? Well, it's 1 times 10 to the minus 2 meters is a centimeter. Okay, so you're talking here a very, very narrow diameter. Okay, if we were to try and convert this to inches, right, 100 micrometers times 1 meter over 1 times 10 to the minus 6, or to the 6th, sorry, micrometers. And then we'll convert this to centimeters, right? There's going to be 100 centimeters in a meter. Okay, and you'll see here micrometers cancels, meters cancel. Okay, and you're left with centimeters. So that works out to 1 times 10 to the 4th over 1 times 10 to the 6th centimeters. Okay, and what's that equal to? Well, you can cancel here, right? That becomes 1, that becomes 2, okay? So that's 1/100th of a centimeter. Give you an idea again of the diameter. 1/100th of a centimeter. Remember that 2.5 centimeters makes an inch. Okay, so you got some really long but relatively small diameter cells making this up. Okay, they have multiple peripheral nuclei. They're surrounded by a plasma membrane that's called the sarcolemma, and they're filled with a semi-solid liquid which contains proteins and ions and waste products and fuel, and also a bunch of organelles we learned about in the cell chapter, and that's the cytoplasm of the muscle cell, called the sarcoplasm. You'll also find glycosomes, which are there for glycogen storage. Glycogen is simply a sugar polymer that serves as an energy storehouse for the muscle cell, and then myoglobin, which is present for oxygen storage. Okay, the muscle cell needs that because it needs quick energy in the event that it's performing work quicker than the blood can deliver fuel, and it needs oxygen for the same reason. Sometimes you'll exercise so hard that you'll temporarily run short of oxygen and fuel, and what these structures do is sort of bridge the gap. Okay. Modified structures include myofibrils, which are basically stacks of muscle protein inside the muscle cell, the sarcoplasmic reticulum, and also the T-tubules. Okay? The sarcoplasmic reticulum, as we're going to find out, is a modified endoplasmic reticulum, which primarily is tasked in the muscle cell with storing calcium, and the T-tubules are deep invaginations of the muscle cell membrane that allow the electrical current that's produced by the release of neurotransmitters by the neurons that talk to these muscle cells to be carried deep inside the muscle cell and begin the process of contraction. Myofibrils are densely packed rods that are inside a single muscle cell. They make up 80% of the cell volume, and they contain contractile units that are called sarcomeres, which contain myofilaments. Each exhibits striations. These are perfectly aligned, repeating series of dark and light bands. The dark bands are called the A bands, and the light bands are called the I bands. Now, something that's very interesting about this muscle cell, this is just a segment of a single muscle cell here. You notice that there's more than one nucleus in a skeletal muscle cell, and the reason for that is when muscle cells are formed, they form by muscle cell progenitors gathering in a group and fusing together, losing their intervening cell membranes and becoming one long cylindrical cell, whose job it is to make tons and tons of muscle protein and, of course, convert energy into movement. Okay, these striations are an indication of the regular arrangement of these muscle proteins in the myofibrils. The H zone, known as the hypochromatic zone, meaning it is less dark than the surrounding bands that it neighbors, is the light region in the midsection of the A band where the filaments don't overlap. The M line is the line of protein made up of myomesin that bisects the H zone and essentially serves as an anchor for the myosin. And the Z disc is a coin-shaped sheet of proteins on the midline of the I band that anchors the thin filaments and connects the myofibrils to each other. Okay, so what you have here is an interdigitation of what we would call thick filaments, which are primarily myosin, and thin filaments, which are primarily actin. Okay, the thick filaments run the entire length of the A band, while the thin filaments run the length of the I band and partway into the A band. The sarcomere is simply the region between two successive Z discs. So in this diagram, the sarcomere would run essentially, and let's use blue for this, from this region, okay, this region here, over to this region there. Okay, so we'll say one sarcomere. Okay, and what happens when the muscle contracts is that the myosin, which you can see here, okay, the red lines here, is going to reach up and grab the actin, which are the gray lines, and is going to pull them inward and cause the entire sarcomere to shorten, and that is going to result in the entire myofibril shortening, which will result in the muscle cell shortening, which will also result in turn in the muscle shortening. Yeah, the sarcomere is the smallest contractile unit of the muscle fiber. It aligns along the myofibril end to end to end. It contains an A band with half an I band at either end. It's composed of thick myofilaments made of contractile proteins. The main protein, of course, is myosin. That's the thick filament. Okay? And you can see how in the myosin we have these little protrusions, these golf ball head shaped globular regions. What they do is they actually do the reaching up and the grabbing of the actin when the muscle gets ready to contract. The myofibril banding pattern is essentially an orderly arrangement of actin and myosin in the sarcomere. The actin are the thin filaments, and they extend across the I band and partially into the A band. They're anchored to the Z discs, and the myosin myofilaments are the thick filaments that extend the entire A band and are connected at the M line. If we look at the thick filaments in detail, what we see is they're made up of a polymer called myosin. Each is composed of 2 heavy and 4 light polypeptide chains. The myosin tails contain heavy interwoven polypeptide chains. They are dimers. The heads contain 2 smaller light polypeptide chains that act to cross-bridge during muscle contraction. These are binding sites for actin in the thin filaments. And they also contain binding sites for ATP. This is critically important because the ATPase that's contained in these thick filaments is going to be used in order to basically hydrolyze the ATP, release the energy from that molecule and trap it, and then convert that into energy for contraction. Okay, the thin filaments are twisted double strands of actin. The actin consists of G-actin subunits. When the G-actin subunits connect to each other, they form what we call F-actin. G stands for globular, and F stands for fibrous. So that's where the names come from. The G-actin has active sites for the myosin heads to attach during contraction, and tropomyosin and troponin are the regulatory proteins that are bound to the actin. And what they do is they serve to regulate when the muscle is going to contract. The troponin you could think of as a handle or a pivot point. What happens when muscle contraction begins is that calcium will bind to troponin and cause the tropomyosin that it's attached to pivot out of the way between the myosin and the actin so that cross-bridging can actually occur. When the muscle is relaxed, the calcium is released from the troponin. This causes the tropomyosin to pivot back in between the actin and myosin, and the muscle goes back to its resting length. So what you're looking at here is just the ultrastructure of the thick and the thin filaments. The main thing I want you to pick up with the thick filaments are these myosin heads that are capable of reaching up and grabbing the actin and pulling on it, and that's what produces the muscle contraction. And then if you look here at the long polymer, that's going to be attached ultimately at the M line, okay, as an anchor. And these guys are going to serve like ratchets. So they'll reach up and grab and pull, reach up and grab and pull to the actin which surrounds them, okay? Notice that the actin is a polymer. The tropomyosin is the block between the myosin and the actin that inhibits actin-myosin cross-bridging, doesn't permit contraction when it's in the way between actin and myosin. And the troponin are these little yellow areas here, and when they bind calcium, they cause this entire assembly to pivot, to rotate on its long axis so that the myosin can reach up and grab the actin. Okay, there's another type of filament that you find in the myofibril, and it's composed of a protein called titin, which holds thick filaments in place and helps recoil after stretching. It resists excessive stretching, okay? So the titin is sort of an anchor for the thick filaments. Dystrophin links the thin filaments to the proteins of the sarcolemma. Okay, remember the sarcolemma is the muscle cell membrane. So this is how we transfer the force that's produced by the filaments sliding over the top of each other to the muscle cell membrane, ultimately to the connective tissue that holds the muscle cells together, and then from the connective tissue to their attachment to bone or to whatever it is that they're going to move. You've probably heard of a condition called muscular dystrophy. Certain forms of muscular dystrophy are characterized by defective dystrophin. This results in the muscles contracting but not producing movement because they're not effectively anchored to their origins and insertions. The result over time is that if the muscle cannot exert force, it will eventually form contractures, and then eventually the muscle will begin to atrophy. Because in order for muscle to remember to make muscle protein, it has to be called into action as muscle tissue. If contraction doesn't occur frequently enough, the muscle tissue quits making muscle protein. Eventually the muscle cell disappears in a process known as atrophy and is replaced by scar tissue. Forms of muscular dystrophy can be severe or mild. Some of the more severe forms primarily affect men more than women because the defective gene is carried on the X chromosome. Remember that in men there's only one X chromosome, so any bad information that shows up on the X is going to be characterized as the disease condition in the male, whereas in women with two X chromosomes you have a second X that can essentially cover for bad information in a defective gene on the other X chromosome, and so you don't see these X-linked conditions showing up nearly as often in women. Okay, we call these kinds of disorders sex-linked because they're associated with the X chromosome. The Y chromosome, which is only present in men, only has one trait on it, and that's the trait of being male. So anything else that's on the X is going to show up. So guys, if you have a sex-linked problem, sex-linked disease, examples include color blindness, hemophilia, and male pattern baldness, don't look at your dad, look at your mom, because that's where you inherited it. Okay, nebulin, myomesin, and C proteins bind the filaments of the sarcomeres together, and they help maintain alignment. The sarcoplasmic reticulum is a network of sacs that contain large amounts of calcium inside the muscle cell. We call it sarcoplasmic because it resides in muscle, but essentially it would correspond to the ER in a normal cell, in some other cell type. It has to run longitudinally and also vertically throughout the muscle cell in order to deliver its calcium payload when it's electrically stimulated. There are pairs of terminal cisterns that form perpendicular cross channels. Now, you might say, what in the heck is a terminal cistern? Essentially, this is a swelling that's at the portion of the sarcoplasmic reticulum that's nearest an invagination of the cell membrane called the T-tubule. Okay, and we'll see that it's these structures, the T-tubules and the terminal cisternae, that form the initiation of calcium release inside the muscle cell that ultimately leads to contraction. It functions in the regulation of intracellular calcium levels, and it stores and releases calcium. So the T-tubules are continuations of the sarcolemma that dip deep down inside the muscle cell so that we can carry the electrical signal into the core of the skeletal muscle. The lumen is continuous with the extracellular space, and they increase the muscle fiber's surface area. They penetrate the cell's interior at each A-band, I-band junction, and you can see them here, the invaginations, the T-tubules. And they associate with paired terminal cisternae to form a structure called a triad that circles each sarcomere. So you can see here this blue bulge, this white T-tubule, and the other blue bulge. That constitutes a triad. T-tubules conduct impulses deep in the muscle fiber to every sarcomere, and there are proteins that are embedded in the intermembrane space from the T-tubule to the sarcoplasmic reticulum, and they connect to each other. The T-tubule integral proteins are voltage sensors that change their shape when the voltage of the membrane changes, and this results in calcium release inside the cell that's ultimately going to result in the contraction of the muscle cell. So how is it that we generate the force in muscle cells? Well, we use what we call the sliding filament model to explain how the force is generated. It doesn't necessarily cause shortening of the fiber, but it does produce an increase in force. Shortening occurs when tension generated by the cross-bridging on the thin filaments is greater than the forces that oppose the shortening. In a relaxed state, the thick and thin filaments overlap only at the ends of the A band. In the sliding filament model during contraction, the thin filaments slide past the thick filaments as the thick filaments reach up to grab and pull on them. And this increases the amount of overlap between actin and myosin. This is what we call cross-bridge formation, and it's a repeating cycle of binding, pulling, releasing, and binding again further down the actin filament so that we can pull the ends of the sarcomere closer and closer together. The myosin heads bind to the actin and begin the sliding. The cross bridges form and break many times, and this ratchets the thin filaments towards the center of the sarcomere, and this causes it to shorten. It pulls the Z discs towards the M line, and it increases, of course, the overlap between the thick and the thin filaments. So what you'll see is the I bands shortening, the Z discs getting closer together, the H zones disappearing, and the A bands moving closer to each other as well, while the length of the A band stays the same. And so you can see here a relaxed and a contracted skeletal muscle fiber. So notice the different bands, okay? Notice how the overlap between the thick and the thin filaments is minimal in the relaxed muscle fiber. You can see here the edges of the A band overlapping the edges now of the thin filaments, okay? The area of overlap is essentially this dark region that's on either side of the H zone, okay? And then what happens as we contract is that the thin filaments are pulled towards the center. The degree of overlap between the thin and the thick filaments increases. The H zone will shrink, and also the I bands will shrink. Okay, so notice how the I bands got smaller. See how the light region is very thick here and very thin here. And then see how the degree of overlap, all right, increases as the muscle pulls the elements of the sarcomere closer and closer together. The H zone begins to vanish, the I zone begins to vanish, and the amount of overlap increases. The length of the A band does not change, okay? The location of the M line does not change, all right? But the Z discs draw closer together, and the hypochromatic zone vanishes, and the I bands become thinner. For skeletal muscle to contract, we need activation at the neuromuscular junction, and this results from nervous system stimulation. It has to generate something called an action potential in the sarcolemma, which is going to lead eventually to excitation-contraction coupling. The action potential gets propagated along the sarcolemma, and intracellular calcium levels rise. Now you might say, okay, well wait a minute now. What is an action potential? Well, an action potential is the biological equivalent of an electrical current. And the way that an action potential operates is by changing the separation of charge in a particular region of the membrane. The membrane starts out in what we call a polarized state. Okay, so if we were to sketch this, we'll use here, let's use red for this. Okay, so if we were examining a region of the membrane, okay, so this would be the cell membrane. Okay, this would be a muscle cell, a section of cell membrane here, or I'm sorry, nerve cell. And what we would have on the inside of the cell is a negative charge, and on the outside of the cell we would have a positive charge. Okay. That state exists because of the action of the sodium-potassium pump, which sends 3 positive sodium ions out of the cell for every 2 positive potassium ions that it pumps into the cell. And in addition to that, we have leakage across the cell membrane, but the leakage is kind of in one direction. The membrane is much leakier to sodium or to potassium than it is to sodium, and the result then is that sodium tends to be isolated outside the cell, and potassium tends to be isolated inside the cell, but there's also less positive charge in the cell than outside. Okay, now, so that's the polarized state. So we'll call that the polarized state. Polarized. Now, what happens when a chemical signal is produced by a nerve cell, or by a, let's say a chemical signal that attaches to the membrane here. We could consider this to be a nerve cell, or in some cases a muscle cell. Doesn't matter. We're just looking at the phenomenon of an action potential here. What happens is that there are certain gates that will open up. We call these ion gates. We'll draw them here in black. And when these ion gates pop open, after they bind to their chemical signal, what will happen is they'll start letting sodium into the cell. Okay, so sodium flows from the outside to the inside. The result is that that area of the membrane will become depolarized. Depolarized. Now, what happens next is that eventually what's going to occur is that the voltage is going to change to the point in that region of the membrane that those sodium channels that were initially open will shut, and then potassium channels will open up, and then potassium will rush out, and we'll indicate that as, say, in blue. Okay, so let's do that in blue. When the potassium starts to rush out, what will happen is that the membrane will go from a depolarized state back to a polarized state, and even to a hyperpolarized state. And then the membrane will be restored to its resting membrane potential by the action of the leak channels and the sodium-potassium pump that's always operating in the background. Now, as this change in membrane potential spreads, what happens is that it opens up sodium channels in neighboring regions of the membrane and starts the whole process over again in another area, and then that entire cycle repeats. And so what will happen is that the action potential will proceed in one direction, at one speed, and at one magnitude from where it's begun to where it will eventually terminate. In a nerve cell, it'll stop at the end of a long process called the axon. In a muscle cell, it stops once we spread all along the length of the muscle cell and down the T-tubules. Okay? So it's sort of like playing dominoes. Once you tip that first domino over, the others are all going to fall. The same thing is going to occur here with an action potential. And that's what we mean when we say that this is sort of like electrical current in living systems, okay? So it's a little bit different than electrical current that runs through a wire, which is essentially electron pushing from one place to another. Here we're looking at changes in the location of charged particles called ions, and those changes move in a wave from one point to the next all the way down the cell membrane. Okay? And the only reason this can happen is because of the location and the density of the pumps and the channels all along this region of the cell. Okay, that's what allows this to occur. So what we're looking at here is just the series of events that leads to muscle contraction. The action potential arrives at the axon terminal at the neuromuscular junction. The acetylcholine is released and binds to receptors on the muscle cell membrane. That's the sarcolemma here. Okay. Ion permeability in the sarcolemma will change, and this will cause a local change in the membrane voltage, which produces a depolarization. The depolarization, which occurs at an area of the muscle cell membrane called the end plate, which lies just underneath the terminus of the axon, starts the action potential in the sarcolemma, provided that the stimulus from the nerve cell is sufficient to generate the action potential. In order for that to happen, we have to reach something called a threshold voltage. At that point, the action potential travels across the entire sarcolemma and down the T-tubules, causing the sarcoplasmic reticulum to spill calcium into the cytoplasm of the muscle cell. The calcium is then going to bind to the troponin. The troponin, of course, is attached to the tropomyosin, which is attached to the actin, And what will happen is this will cause a pivot of the tropomyosin out of the way between the actin and myosin, and then cross-bridging can occur. So skeletal muscles are stimulated by somatic motor neurons that we control. The axons of these neurons travel from the central nervous system through nerves to the skeletal muscle, and each axon forms several branches as it enters the muscle. Each axon forms a neuromuscular junction with a single muscle fiber, and there's usually only one per muscle fiber. So what we're going to look at here are the events at the neuromuscular junction.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, and these are the events that lead to the cross-bridge cycle.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross-bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
And what you're looking at here is just a better diagram of what I was trying to illustrate a few slides back. Okay, you can see here the axon terminus of a muscle of a somatic motor neuron over the top of a motor end plate. When the action potential reaches the end of the axon, calcium rushes into the axon terminal, and this results in the fusion of synaptic vesicles that contain neurotransmitter with the presynaptic membrane of the axon terminal. It dumps its chemical communicator out, it's acetylcholine in the case of skeletal muscle, and it walks its way across this little gap, okay, which is called the neuromuscular junction, and then attaches to basically channels, ligand-gated channels, that when the acetylcholine attaches to them, they open up, and the main ligand-gated channel is a sodium channel. Okay, the sodium channel then lets the sodium in, and what happens is that the membrane potential begins to rise. Okay, so it goes away from its resting polarized state towards a depolarized state. So it'll head up from about minus 95 millivolts to 0, and then it'll peak out at around plus 30 millivolts. And at that point, the sodium channels will close and the potassium channels will open. The potassium then rushes out of the cell then repolarizes, goes back to its resting membrane potential, and then we restore the original distribution of potassium and sodium across the cell membrane by the action of the sodium-potassium pump. And at that point, we're ready for the next signal to come from the motor neuron. And this is just showing you the electrical events that occur in the region of the neuromuscular junction. So here we have the resting membrane potential. The acetylcholine binds to the ligand-gated sodium channels. The sodium rushes in, and that area of the membrane depolarizes. We peak out at about plus 30 millivolts. Two things then happen. The sodium channels close, the potassium channels pop open, the potassium rushes out, okay, and the entire area of the membrane in that region repolarizes. We head back down towards our resting membrane potential, minus 95 millivolts. At this point, the potassium channels shut, and then the pump is going to restore the original concentration of sodium and potassium across the cell membrane. So this is just a look at the events of excitation-contraction coupling.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, this is a zoom up showing you what's happening deep inside the muscle cell once we have the release of acetylcholine as triggered by an action potential arriving at the end of the axon. Okay, so again, the acetylcholine crosses the neuromuscular junction. It attaches to the ligand-gated sodium channels. The sodium rushes in, the membrane depolarizes, and then that depolarization wave is going to move longitudinally down the muscle cell, but also deep inside these imaginations called the T-tubules. Once that occurs, we're going to have the release of calcium inside the muscle cell as these integral membrane proteins that span the T-tubule and the sarcoplasmic reticulum start pumping start releasing the calcium out. It's a passive movement out because there's more calcium inside the sarcoplasmic reticulum than outside. As the calcium floods the sarcoplasm, it binds to the troponin. The troponin causes the tropomyosin to pivot out of the way and exposes the myosin binding sites on the actin. Myosin, in the presence of ATP, can then reach up, bind to the binding site on the actin, and using the ATP for power, basically pull on the actin filament, and that produces the contraction. The cycle repeats over and over again until what happens? Until we quit getting the electrical signal from the motor neuron. So nerve impulse reaches the axon terminal. The voltage-gated channels let the calcium into the terminus of the motor axon. Acetylcholine is released in the synaptic cleft as the synaptic vesicles fuse with the presynaptic membrane. The acetylcholine binds to its receptors on the sarcolemma. This opens ligand-gated sodium and potassium channels. This causes an end plate potential to be produced. If the end plate potential is sufficient, what will happen is voltage-gated sodium channels will pop and the action potential will move longitudinally across the muscle cell and then deep into those T-tubule invaginations. The voltage-sensitive proteins in the T-tubules change their shape, and we have release of calcium from the sarcoplasmic reticulum into the cytosol. At low intracellular calcium concentration, tropomyosin will block the active sites on the actin, so the myosin heads cannot cross-bridge, and the muscle fiber relaxes. At high calcium concentrations, the calcium binds the troponin, which changes the shape and moves the tropomyosin out from between the actin and myosin. The myosin heads then can bind to the actin. This causes the sarcomere to shorten and the muscle to contract. When the nervous stimulation stops, the calcium is pumped back into the sarcoplasmic reticulum. The actin filaments pivot so that the tropomyosin is in between the actin and myosin. Then the muscle myofibrils go back to their resting length, and the cycle is complete. And what this is just showing you here is how it is we engage in cross-bridging and attachment, power stroke, and then detachment. Okay, so we start here with assuming that the tropomyosin has been removed from between the actin and the myosin. What happens in the presence of ATP is a cross-bridge formation will occur. Okay, and when we bind the ATPase in the head will hydrolyze the ATP. At that point, what will happen is that the chemical potential energy will be converted into kinetic energy. The power stroke will ensue. The myosin heads will pull the actin filaments towards the center of the sarcomere. All right, and then it's only when another molecule of ATP attaches to the myosin head that it can detach from the actin and repeat the entire process further down the actin filament. And so the whole thing repeats and repeats over and over again until, of course, the nerve signal from the motor neuron ceases. An interesting outcome of this is the phenomenon of rigor mortis. Back in the day, when we didn't have embalming, and the wild, wild west was truly wild, wild, the fastest gun in town was the sheriff. And so what you do is you face down at high noon in the middle of Main Street, and whoever was quickest on the draw, they would be the next sheriff. Well, what happened to the loser in the gunfight is that we would have a funeral within a day or two, and they'd put the body in a pine box, and often in the middle of the funeral, the body would sit up and would scare the bejesus out of everybody who was in attendance. This was a phenomenon known as rigor mortis. What happens here is that we have cross-bridge detachment failing to occur because we've used up all the ATP in one final contraction. 3 to 4 hours after death, the muscles stiffen with weak rigidity at about 12 hours post-mortem. The dying cells take in calcium and cross-bridge formation occurs. No ATP is generated to break the cross bridges, and so the muscles remain in a state of tension. Okay? We don't have that anymore because now we embalm the bodies and we, of course, clip critical muscle attachment points so that this doesn't happen during a modern-day funeral. But back in the day, it was a common occurrence. Now, this is also a useful tool in determining time of death. There's a very famous location at the University of Tennessee called the Body Farm. And what happens at the Body Farm is people donate their bodies to science after they die, and they put them outside in the elements. And what the students who are enrolled in the forensics course do is they go out on a field trip, they find the bodies, and they use, among other things, rigor to determine the time and the cause of death. Okay, so a lot of other clues that they can put together in order to determine the time and cause of death. Rigor just happens to be one of them. Now, eventually, what happens is the rigor will cease as the muscle proteins begin to decompose. That results in detachment between the actin and myosin, and the muscle goes flaccid. Okay, so it's not a permanent condition. It eventually disappears. Okay, that brings to the end this chapter. We'll pick up with 9b in class next time we meet, and I'll see everybody soon.